University of Belgrade.
Oh, okay, good. All right.
So we are close.
Okay.
Okay. Just start the recording. Record to the cloud. Okay, everything should be set now.
I gotta hit. Got it. Okay.
Okay, great. So, good evening, everyone. It is our great honor to host today Bruce Albert. He's a biochemist, and he's committed to the improvement of science and mathematics education. As we have found out recently, he supports and he always accepts invitations from students, and that's why he's here today, and we are thankful. He was awarded the National Medal of Science by President Barack Obama in 2014 and the 2016 Lasker Special Achievements Award in Medical Science. Bruce served as editor-in-chief of Science and as one of the first three United States science envoys. He is now the Chancellor Leadership Chair in Biochemistry and Biophysics for Science and Education at the University of California, San Francisco, to which he returned after serving two six-year terms as the president of National Academy of Science. And he's also the most known for the book which I'm holding. And it's one of my favorite book in molecular biology. So thank you very much for being here today with us. And the floor is all yours.
Thank you, Todor. It's a pleasure to be here. I just want to start by showing up. Let's see if I can get this thing to work. Okay, now this is, okay, there we go. This is my close friend, Vladimir Glisson. I was a graduate student at Harvard with Paul Doty, and there were four research benches in the lab that I was in, and Vladimir and his wife Mira occupied one of them, and he was a postdoc, obviously from then Yugoslavia, and later he would go back. He got in trouble with Milosevic, had to come to the United States for a while, but then went back, and he was a founder of your IMGGE, which I have visited, and when I visited him in Belgrade. So I wouldn't call myself an expert on Serbia, but I at least have been there several times. Thank you. All right, so this is an outline of what I want to say. I want to talk a little bit about what I did when I was a scientist. And I'm going to talk a little bit about the textbook, which has a lot of lessons for our scientists, I think. But then, of course, I'm going to emphasize throughout what I've learned about doing good science. And hopefully we can have a discussion afterwards. I'll try to talk for only about 35 minutes. So I'm going to start with my life in science, and subtitle would be The Biochemistry of Protein Machines. When we started, I knew nothing. Nobody ever suspected there were such a thing as protein machines. I was a high school student when this inspirational breakthrough, the discovery of DNA double helix by Watson and Crick occurred, and that was the inspiring moment for really what started what we now call molecular biology. It set a interesting tradition at the time. We thought that everything would turn out to be relatively simple in biology, in cell biology, because the DNA double helix itself was so elegant and straightforward. Of course, it's turned out to be much more complicated than we had imagined. So Watson and Crick, in theory, in their short papers, solved the mystery of heredity, but they didn't know anything about biochemistry. And in there, if you read their paper, they even imagine that DNA replication might occur without an enzyme. But starting as a graduate student in 1961, I spent a lot of years working on trying to unravel what the machinery was for DNA replication. And I made a lot of mistakes. One of the themes of this talk is going to be we all make huge numbers of mistakes. learning from failure is an important part of life. And that's what makes some older people good mentors, because they've failed so many times, they've learned a lot from their failures. Of course, others don't learn anything from their failures and they keep on making mistakes, and that's not good. So try to study all of your failures carefully and learn from them. So this is how we thought DNA replication worked. An amazing enzyme, DNA polymerase, which you all know about, had been discovered by Arthur Kornberg. It would earn him the Nobel Prize shortly later. And we thought that this one enzyme was all there was to replicating DNA. And we thought that, the template specified addition of one of the four nucleotides at a time was something that occurred through a random collision process. The DNA polymerase would bind to the 3' end of a growing strand, add a nucleotide, fall off. The next DNA polymerase would come in, collide, add a nucleotide, and fall off. And we thought this was possible because the speed of molecular collisions in the cell is enormous. It's hard for students to imagine really how fast things are interacting and binding. So as a physical chemist in the lab that I was in, we all thought that we didn't need any kind of higher organization, that the cell would be like a test tube full of very concentrated molecules all randomly colliding with each other. Of course, that was wrong, and I'll emphasize that. So I had a dismal career as a graduate student. I had my own theory about how DNA polymerase might be able to replicate a double helix, which was a form of DNA in the cell, even though in a test tube it would only work on single-stranded DNA. I did many experiments to see if my theory was right, and of course it wasn't right, and the no answers did not surprise anyone and did not add to the store of scientific knowledge. Nevertheless, my professor decided, it was time for me to get a PhD, so I wrote up all my field experiments. All I had to do was walk into a room with a group of famous Harvard professors. They talked to me for an hour, and then I passed. That always happened. But to my great surprise, they decided they would fail me because they didn't like my thesis. And I wrote this up as one of many examples of how one learns from failure. And in retrospect, that failure really shaped my whole career because I thought completely differently about how to do science. And basically, the lesson was that theoretical biology is too difficult. The cells are not really organized necessarily according to what you might think and make up. A cell arrived by a very sloppy process of evolution and natural selection. So it hasn't been engineered to be efficient. It just works. And so you have to figure it out by experiments. And in experiments, having a good strategy is the key to success. I had just been making lots of measurements that weren't very meaningful, but I often talk to students about science being a very creative endeavor. Think of it like painting a beautiful picture. A famous painter like Rembrandt had incredible talent in thinking where to put the paint. And if you gave me the same paints, I would just make a mess. Scientists, likewise, are very creative. You have to think carefully about what you want to do and find a strategy that makes sense so that whatever you find, you'll make some progress. The way I did my PhD research was a very bad design. So finally, I did get my PhD 6 months after I failed. I did more experiments, and I went off to Geneva as a postdoc. And I immediately discovered that DNA replication requires much more than one enzyme. And that's because one of my colleagues there, Richard Epstein, a senior professor, had published all this genetic analysis on a model system that started molecular biology, bacteriophage T4, is a very large bacteriophage with a double-stranded linear genome, and it makes all its own replication proteins. And the genetics had shown in a paper that was published right in the middle of my PhD that I'd never read, that there must be at least 7 proteins needed to replicate DNA in addition to DNA polymerase. And so the lesson learned is much easier to do today. Read very widely in the scientific literature so you don't get on a wrong track without knowing some important piece of information for what you want to do. Okay, then I started, after a year, I had a job as an assistant professor at Princeton University, and I had young undergraduate students and graduate students, a few of them, and we all worked together trying to purify each of the T4 bacteriophage proteins that had been identified by the 7 mutant genes. And we had this dream that someday we'd have all these 7 proteins, we mix them together with double-stranded DNA in a test tube, and we get double-stranded DNA replicate. And we had a great day. 9 years later, 1975, it finally happened. This is a picture of my lab back in 1969, is it? I can't really read it. But anyway, that's me in the red shirt with suspenders. And there's several undergraduates, exactly your age, and a couple graduate students. The dog was not part of the lab. It just showed up for this picture. And here's what we discovered. We discovered a protein machine that, like a sewing machine, that replicated DNA. And the whole process is complicated because the DNA double helix is anti-parallel, so that and DNA polymerase only goes in one direction. So one strand could be made continuously, called the leading strand at the bottom, but the top strand has to be made into a series of short pieces going backwards and then stitched together. Nobody would have ever predicted this mechanism. If somebody had said, this was what the theory of how DNA replicated in 1961, we would all said they're crazy. It could never work. All these pieces would fall off. It would never be able to work. But indeed it does. And the magic of protein machines, which now underlie all of cell biochemistry, is best appreciated by the movie that shows this machine in action. This is a little movie that we have associated with our textbooks, but it was made by Cold Spring Harbor Lab to celebrate I think it was the 50th anniversary of the DNA double helix. I'll just play this one briefly.
Using computer animation based on molecular research, we are able to picture how DNA is replicated in living cells. You are looking at an assembly line of miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called a helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into 2 strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is 2 new DNA molecules.
Of course, this is my favorite movie, but at any rate, it really shows how all of life works. The chemistry is incredibly sophisticated, the most sophisticated chemistry by far that we know about. So here's some lessons learned. Because of evolution, there are remarkable homologies between living things. Therefore, use model organisms wherever possible. We use T4 as a model for studying how replication works. And that and other work on simpler organisms was the shortcut to understanding how humans replicate DNA. And we use many more proteins. It's much harder to work out from scratch, but it's basically the same mechanism. And so model organism findings form the basis for then understanding more complex organisms like ourselves. Secondly, nearly all cell processes will be driven by 10 to 20 proteins organized as a protein machine and incorporating ordered protein movements driven by the energy of ATP, hydrolysis. The hydrolysis of tightly bound ATP to ADP drives shape changes in the protein, allosteric changes that give it a directional movement, just like electricity drives the machines in one direction that we know about, that we use in our everyday life. And finally, that all the things that we want to understand are based on elegant mechanisms that are too complex to predict. Okay, now I'm going to change topics to another near failure in writing a textbook. We had to learn from all the mistakes we made as we were trying to do it. This is what happened. In 1978, I'm sitting in my office and I get a call from Jim Watson. And he says, oh, we're going to write this new, he had a beautiful insight that there's time to unite a field called cell biology, which at that point was all about images, light microscope and EM images, with the new field of molecular biology, which I was working in. And he says, it's only going to take 2 summers to write this textbook, and next summer and the summer afterward. And so the second summer I worked on it was 1979, when we were supposed to finish, and we barely had started. And it was almost, we almost decided to quit. This is what it was like to write a textbook like ours before laptop computers. You type it out, you write it out longhand, somebody would type it out, somebody would mark it up. I can't remember. This is Julian Lewis's chapter on development. It's marked up. My pen is my writing, and somebody else's writing is the other one. So this is how we write our textbook, but now we do it in computers, of course. Finally, by 1981, we realized that we could actually do this, and the first edition was finally published 2 years later. And this is the cover of the first edition, and I just want to emphasize that it took us over 365 12-hour days working together to produce this textbook. It was an enormous amount of effort. We all learned a huge amount by doing so. And this is a funny picture of us crossing Abbey Road, where we often worked in London because most of the authors were from the UK, and we worked in a house near Abbey Road. So this took this photograph as a joke. So each time we write a new edition, this is the latest one, this came out, we're humbled by how much we don't know. And this is really important for young scientists to know there's a huge amount for you to do. So here's some of the things that we only realized fairly recently. First is that extensive sets of scaffolds, special protein and RNA molecules, produce biochemical subcompartments in the cell without requiring a membrane. That many of these scaffolds can assemble to create larger phase-separated liquid compartments, and that this kind of organization is critical for life. And second, I'll talk about the fact that given all the complexity, we can't really understand what's going on right now without new methodology. So scaffold proteins, here's a basic idea from our textbook. You could speed up specific reactions and localize them in a specific part of a cell by having a gray molecule like this scaffold bound to some certain part of the cell. It binds these colored protein molecules or RNA molecules. They're going to react with each other, and it greatly increases the rate at which they collide, and therefore it both speeds up specific reactions and it localizes those reactions in specific parts of a cell. And the simplest example I know of a scaffold is the little scaffold on the protein called formin, the green protein, that has these whiskers that carry new monomers of the protein actin that forms a major filament in the cytoplasm. The filament is this red thing that would go down many stories through the floor. This is showing the very end of it. And the end grows at enormous rates because of the scaffold that are holding the precursors, the little red molecules, close by so that they can collide really fast to make the end grow. And that is important for enabling cells to be effective. But that's just one of many examples. RNA scaffolds are also widely used, the so-called non-coding RNAs, most of which we don't understand really yet. And here's this an old picture of a cell nuclease showing that, when you make RNA, so-called transcription, the proteins that needed for catalyze the RNA synthesis reaction of transcription get concentrated in these little blobs. And then when you make DNA, you have a DNA replication factory where its proteins get concentrated in other blobs. Those are green. Importantly, these form and come and go as DNA replication starts and stops. They're very transient structures that, greatly accelerate and make possible the biochemistry by making the many proteins that are needed for each reaction highly concentrated where they're needed. And we now call these biomolecular condensates, and they form many different membranous organelles. The nucleus is the most famous one. It's where ribosomes are made, this big thing, but there are many other such structures. We're just learning more about them. They were surprised, as maybe 10 years ago, we didn't suspect that they existed. In conclusion, a cell is nothing like a test tube. Nearly everything is organized inside the cell by protein and RNA scaffolds. So the way we thought about it in the early days was completely wrong. Okay, the second new idea that is given the name emergent properties. Emergent properties is a name invented by physicists in about 1940 to describe the fact that when you have many complex interactions, they create something that's different from, qualitatively different from what the individual components are. And this happens all the time in living systems. And we don't really have adequate tools to decipher them. And that's the point of this very brief survey. I'm going to say, when I was a student, we learned that A goes to B, goes to C, goes to D. But now we know that that wouldn't work without this feed forward and feed back loops that make it useful to the cell. All these reactions have to be carefully controlled in very complicated ways. And even this simple example, there's no way to understand it without mathematics. Here's a much more realistic example of what actually is going on. This is the actin cytoskeleton. That's the same protein I talked about that was red, now it's gray. And the molecule that controlled its growth are those molecules, so-called formins, are colored purple in this diagram. We're showing an interaction. All the different actin binding proteins that make actin effective for the cell, that make the cytoskeleton work for the cell, make different structures at different parts of the cells, depend on all these different interactions. The famous protein myosins are green. That's the motor proteins that make things move along actin filaments and so on. This is, this the yeast cell. It's even more complicated than the human cell, but even when we know all these interactions and all their binding constants, all the structures, it's really, you don't have a way of understanding it. So even when we have a complete knowledge of all the molecules, protein machines, and molecular interactions in a cell, we will not be able to understand even the simplest of the living cells. Instead, life reflects the emergent properties that result from very complex networks of chemical interactions. So this is sort of my basic conclusion. We're going to need much more biochemistry. Young people, we need an army of young people to do reconstruct purified systems the way we did with the replication apparatus and really come to understand how they work. A lot of the protein machines, we know what their components are, but we don't understand how they function. Also badly needed are new quantitative methods for analyzing and understanding the enormous complexity of life's chemistry. This is going to involve, of course, computer modeling and mathematics. So I just want to end with a current problem in the US, and I doubt that it's not a problem everywhere. So that, those who support biomedical research often do so because they want to cure diseases that they or their relatives have had or their friends. And they don't recognize, and you can't expect them to recognize, that we don't have the abilities to do all the things we would like to do with human disease prevention and cures. And we won't have it until we have more fundamental knowledge about how cells work. And that's not easy to get governments to support adequately, because there's, everyone wants a short-term cure to some disease. This is my favorite editorial from Science. When I was editor of Science magazine for 5 years, I recruited about 250 editorials and wrote some of them. But this is my favorite. Huda Zogby is a wonderful scientist who started as an MD, and she recognized she couldn't solve the problem of these kids that she was seeing with so-called Rett syndrome. And she started to become a scientist, and she became a terrific scientist. But she wrote this editorial for me, bemoaning the fact that not enough fundamental research is done to enable her to do her work effectively. And she used this wonderful analogy that trying to understand her disease is like trying to understand a text with the depth of Shakespeare using a first grader's vocabulary and experience. We need, from basic research, to enable, create the tools of understanding to attack disease and prevent disease much more effectively. So this is just some example of how little we know. There was this very big tour de force in which a minimal bacterial genome was constructed, and it was, to everyone's surprise, about 1/3 of the genes that were required for this bacteria to be able to replicate itself with a division time of 3 hours or less. 1/3 of those genes, we had no idea what they did, and we still don't have an idea of what most of those genes do. So even the simplest cells, we don't understand in the real sense of understanding. That's because of all the emergent properties, in part, that we don't know how to deal with. Another example is the importance of a model organism called Drosophila, the fruit fly. We're still far from understanding how cells work together to talk to each other or form and maintain tissues without giving us cancer. When I taught cancer to medical students at UCSF, I used to say the really amazing thing is that we don't all die of cancer before we're born, because it's so difficult to control all these cells. Many examples show that first working out a mechanism in the fruit fly provides a shortcut to understanding humans. And in our last edition, I have to look for the new edition, the chapter on the development of tissues contained 50 references to the fruit fly, 4 times more than to the next most cited organisms, the mouse. Without the Drosophila work, we wouldn't know almost anything about human development. Consider the human brain. The human brain has the ultimate emergent property of human consciousness. So if you want to understand the term emergent property, there's a good one. From a connection of billions of neurons, we get this thing called human consciousness, completely different quality. So here's how complicated the brain is. It contains more than 100 billion neurons, the human brain, each of which on average has to make connections with the thousands of others according to a regular and predictable wiring plan. People who try to understand the human brain have to be very courageous. The Drosophila brain, on the other hand, has 1 million times less neurons than humans. Every neuron has been mapped. You could do genetics, knock out a gene, manipulate the system in ways you can't do in humans. Until we understand the Drosophila brain, we're not going to have any chance of really understanding the human brain. And yet that kind of work is much less supported, at least by the US government. In summary, where is biology going? We now know that the chemistry of life is incredibly complex, by far the most sophisticated chemistry known. Many of the most interesting attributes of life are due to emergent properties that stem from very complicated networks of chemical reactions with consequences that cannot be deciphered from the details of the individual parts alone. And therefore, innovative new methods and approaches will be needed before we can claim to understand even the very simplest living cells. A lot of people talk about the fact that data is not understanding. So much of today's research supported by our government is collecting data with the idea that if we get enough data, we'll somehow understand it, but it's not going to happen. We need new ways of thinking about how to gain understanding. Okay, I just want to end with this one little sidelight on my other life. I shifted to science policy in 1993. This is a meeting in 2002 in a Swiss McDonald's. You can see the cup. There were 15 academy presidents there, and I only took this picture only shows some of them. But the main point I wanted to say is that we all were unhappy with our governments, how they were making short-term decisions, and we could really work together very effectively to try to spread much more logical thinking and understanding across the world. Boy, the pandemic has really revealed to us how badly that is needed and how few people really understand scientific judgments and why they should pay attention to them. The idea that vaccines are safe is considered by many, it's just the dogma of scientists, what scientists happen to believe. And they could believe the dogma of anybody else. There's no understanding of how science creates this very special, reliable kind of knowledge as a community effort that's self-correcting. So we have a huge effort to do. When to go in science education, I'm working on that now, but that's going to be a huge task to really change the nature of science education so that people don't learn primarily science facts, but they learn how science creates reliable information. And this is my last slide from Louis Pasteur. Science knows no country, knowledge belongs to humanity. It's the torch that illuminates the world. Thank you.
Thank you. It was really great. It was really great talk. So thank you. Do we have any questions from the audience?
I'll have to stop my sharing. Okay.
Yeah, no worries. You can raise your hands or type your question in the chat.
I can look in the chat.
Yeah, Ivana said something very funny, similar to the European Union situation.
Okay, what do I mean by strategy? This is critical for young people, right? So it's very easy just to collect data and measure things, right? There's so many, new tools that we could sequence anything, we can measure anything, we could do, any technique on cells and learn something new. Let me just take the example of cancer cells. There's a huge amount of money supporting people to study various cancer cells, and every cancer is a somewhat different disease. And people measure all kinds of levels of RNA, protein, and stuff. It's just not very clever way of figuring out how we're going to deal with that disease and how it originates. There have got to be better ways. And so what I mean, a strategy, I mean, find something to do that will really make a difference. Because, you have one life as a scientist, you might as well do something that's meaningful to contribute. And the game is not just to get publications or get research grants, although that's very important. the research, you have to get funding, but we need wiser funding mechanisms that really reward original ideas. And by strategy, I mean a strategy that's different from what other people are doing already, that has a chance of making a new kind of discovery and making us think differently about cancer. And that may mean, working with zebrafish and setting up a clever zebrafish system. But to do that, you have to really read a lot. You have to think a lot. Before you start doing experiments, you have to really write down why you want to do that. Analyze it, read things. Because otherwise you're going to waste a lot of time. And a strategy is like, you have to fix a roof. You need a strategy for that. But science, you need a strategy too. And it's just too easy just to get a hammer and start hammering. And so for young people, I always recommend that it doesn't matter really what you do in your initial research. Just find a good scientist who's willing to help you learn how to do science by doing it. And somebody who's doing something that is meaningful, and usually it'll be in a model organism, because those are places where you could really use many different tools and have many different strategies and learn how to combine tools in clever ways. Often when you're working with a human cancer cell, there are not that many ways that you could manipulate it. So, there's so many wonderful things that science needs you to discover. And, when you write a textbook, you realize how many things we don't understand. And I encourage you to think carefully about what you're going to do. And when you're trying to learn to do science, it's an apprenticeship. Work with somebody. I usually advise young people to work with a younger scientist, doesn't have a big group, who's working on some model organism or some really clever thing where they're not competing with everybody else doing the same thing. So you have time to carry out your experiments. You're not rushing to publish. Anyway, it's very important that you think about science in a careful way and not think that science is just getting data. There's a hand up. That you want to call?
Hi, hello. So I think that maybe 20 years ago now already, you wrote an editorial about asking, are we producing too many PhDs?
Yeah.
And I guess 20 years later, things are not getting better. So what's your take on producing too many PhDs, too many papers, too many journals?
Okay, so, we need scientists everywhere. The students who come to us at UCSF as graduate students, I view them as a treasure. They are somehow mutant in our society, full of crazy things. They have decided to do science when they're not been distracted by TV and crazy fads. And our job is to train them to think scientifically and learn how to use strategy and experimentation, both in the lab and in their lives. And I've had at UCSF a number of graduate students who've gone on to decide they didn't want to do what we call normal research. They wanted to work on science education, or they wanted to work on science communication. So they went directly from their PhD to work in a museum, to work in a university as basically a teacher. Anyway, these people were very good at science, and they were also very good at whatever they would do. So we need some of our best students to actually go into these other fields where we can effectively spread science. We can't spread science without what I call science adapters. People who are like these, enthusiastic young scientists who go off and work in other places like newspapers or Congress. In our political system, we have a wonderful system that was developed by, in the ages of 70 years ago. It's called something like that. It's called the AAAS Fellowship, which selects something like 200 PhDs every year and pays them to go work in government on policy for 1 year. And half of these people go back to the lab afterwards, but half of them become staff in government. They become leaders of new non-governmental organizations, or that they're spreading science in other ways. And without those kinds of people, we need those kind of people in the political world. Otherwise, the politicians won't have any way of understanding what science represents, or the world in Washington, when I was there for 12 years, if we had a committee in the Congress, important committee that had one former scientist on it as staff, it made all the difference, because that person was the interpreter between the academy and the politicians. That person could tell us what the politicians needed in the way of judgment from the academy. And then when we tried to give them advice, they could interpret that advice in their world. It's sort of like anthropology, we have all these different cultures in our complex societies. And, we need adapters between the two different cultures of the political world and the scientific world. Those of us who are in science, take it for granted that how science works, but those people outside of the scientific enterprise don't understand. We saw that so dramatically in recent years. Tony Fauci, who's a wonderful leader in science, badly criticized because by many people think he should be fired, because at one point he said, I don't think everybody should be wearing a mask. Well, at that point, we didn't know anything about the virus. We had a very low supply of masks, and the masks that we had should go to medical people. So as the situation changed, as we understood more about how the virus spread, and as we had a supply of masks, then he started saying everybody should wear a mask. Well, that's the way science works. You have to be able to change your mind as the facts change. And yet the public is being susceptible to this idea that Fauci should be fired because he said different things at two different times. It shows a completely misunderstanding of how science works. And there's so many examples of that in the pandemic. It's been very disillusioning to scientists, but it's also, in a way, a wake-up call that we have to think differently about how we teach our science courses. I just wrote 2 weeks ago, or a week ago, I had an editorial in Science with a couple other colleagues, basically talking about college biology 1. We have to change the nature of college biology 1. So it's college in the United States, about 1 million students take first-year college biology of some kind. And that may be the last science course they ever take. And if we have them memorize all the parts of a cell, what's the point of that? It's not going to be useful. They'll forget it. They need to understand how science makes judgments, and why the consensus of judgment of science on something like climate change or smoking or vaccines or masks, why they should pay attention. And they don't right now. And if you look at the way we teach most biology 1 courses, but overloaded with facts, you could see that there's no reason why they should learn how science works from those courses. So this was sort of a plea to my colleagues who read Science magazine to rethink how they're teaching. And that's not only true for biology 1, it's true for introductory chemistry, for any introductory course in college. And of course, the other point that's important to make, the way college teaches science defines what that science is at every lower level. So the fact that we teach biology 1 in most colleges and universities has a huge mass of facts to be memorized. distributes down to 12-year-olds who are asked, for example, to be able to write a sentence about the endoplasmic reticulum. Well, they memorize a sentence about their endoplasmic reticulum, but they have no idea what it is. And then they get an A on the exam, but they hate science. They see it's meaningless, and they have no conception of why they should pay any attention to scientists. And probably many of them grow up to be politicians who hate science and who don't believe in scientists. So we have a big job. It's a job for all of us all around the world to rethink and share best practices, how we could redefine biology 1 and, don't have to, even though I love all the parts of a cell, I don't think most people have to know anything about them. They know there's a nucleus and a plasma membrane and a cytoplasm. That's enough. But they have to understand that the cell is a self-replicating chemical entity, and some sense of how amazing it is, and how cells have to talk to each other in order to know where they are. Was the miracle of embryonic development, to get some feeling about what scientists are trying to understand, but then to be asked to memorize all these science facts about cell biology, it really is not the high priority that many other folks' ways of thinking about biology 1 are. That was a long answer.
Thank you. Yeah. But do you think it's going to be possible to change that paradigm for biology 1 courses?
Well, so in our editorial, which was a week ago, we propose a strategy, which is, first of all, to get groups of scientists to get together and think about, if you only had, 40 lectures, 1 semester to teach biology 1, what would be the most important things to think about? What are the basic principles of cell biology that you want people to know? That's the kind of thing I would love to think about with my colleagues who do our textbook. And if you only had you had 2 weeks to teach everything about cells, what would you do? And then likewise for ecology and the other parts of biology. But then, then you'd have this this is a core what people think. There'll be many different versions of this, but this is a core about what's really essential to learn for a college student in biology. And then you'd be able to make room for the other thing, which is understanding how science works. You need to make time so they can read a few carefully selected original papers and understand how scientists work, to see how science is self-correcting, to maybe use some historical examples of how science has corrected mistakes and gets more accurate with time. At any rate, it's a wonderful intellectual challenge, and it's something that I'm hoping to use Zoom. I think Zoom changes a lot. We used to, in order to get people together, we used to have to fly them all over the place, but we just did an experiment supported by the Gordon Betty Moore Foundation. It's going to be published very soon. A man by the name of Jonathan Osborne from the UK, who's a leader in science education, selected a group of about 10 people from all around the world who were experts in different aspects of science communication and digital understanding and misunderstanding, and to prepare sort of a white paper. It's about 20,000 words about how science education should change in light of all the misinformation that people have to deal with now that we never thought about before, right? When I was a kid, I didn't know anything except watching, Walter Cronkite on, the newscasters on the TV. But now, you've got all this crazy, unfiltered, much of it's created by people who make money by making crazy ideas spread on the internet or on the YouTube, and they get paid by how many, likes they have and how many views by the advertising. I mean, it's just all messed up. So we got a world that's going out of control with magical, irrational thinking. And so this paper was produced by Zoom. So we had these people, 10 people meet 2 hours every, I think it was every third week. And we worked together and produced this white paper in about 6 months, something like that. But we could do that for science education. We don't have to fly people around and waste their time. Obviously, it's better if you could meet with people, but it's so much more difficult. Zoom makes a lot of things possible, including this discussion. And we had these technologies before, but before the pandemic, nobody knew how to use them. But now everybody knows how to use them, so it's effortless to do it. So anyway, I hope that with Zoom also, we can make better connections between young scientists all around the world. I don't know how many of you know about a new movement that I'm very enthusiastic about. It's called the Young Academy movement. The German Academy, in about the year 2000, something like that, the leaders of the German academies got together and did this amazing experiment that everybody thought would fail. They got together something like 100 carefully selected young scientists, that is 35 to 40-year-old scientists, across many different fields, and they define science quite broadly. People work on political science, people who work on communication, but the core were research scientists and engineers. And they said, well, you get together, you decide what you want to do. You have to do something for science for your country, and we'll give you some staff support. They one staff person to help them. Everybody else is a volunteer. And these young academy in Germany just did beautiful stuff, and it was copied then by the Netherlands. They're now, I don't know about Serbia. Do you know? There are 40 countries, 40 nations with young academies. You're only in a young academy for 5 years, and then you're an alumni. Otherwise, it wouldn't be young. But every class gets to, choose their own project. And a lot of people felt, well, old people, well, why should we do this? Young people make all kinds of mistakes and blah, blah, blah. But it was really effective. And of course, the other thing is the young scientists are all over social media, where those of us from the older academies don't have any connection to social media. And so we're, in a way, only the young academies are connected to most citizens in the world. And they've just been incredibly impressive. Oh, so I see Serbia is a member of the Global Young Academy, right? Now we need to get your senior academy interested in to start Serbian Young Academy. All right, we keep working. So I had to work on our academy to start a young academy. The name is, many of them are not called a young academy. They're called, ours is called New Voices. And many of them have other names because the old academy doesn't like to think of themselves as old. So many old academies don't want to support a young academy, but call it something else. But anyway, it took me 10 years before I could get the academy to form, the US Academy to form the Young Academy. And I would suggest you keep the pressure on, because the young academies are, networked all around the world. Serbia needs to be part of this movement. I'm so impressed with what they're doing. Their website is, the Global Young Academy, which is mentioned here. I know, very familiar with that. The Germans have been fantastic because the German government not only supports their young academy, but it supports something called the Global Young Academy, which has a sizable staff supported by the government, that makes this global Young Academy function. And the Global Young Academy website is a great resource for people in Serbia that tells you what other people have learned about how to get their old academies from a Young Academy and how to make sure that the Young Academy works. It's just been amazing. And I've long criticized the website of the Association of the Older Academies, the Inter-Academy Partnership, which I was very active in when I was at the Academy. Anyway, I often criticize their horrible website. And the young people make much better websites. They're much better communicators. Yeah. So at any rate, I know I've been to the Global Young Academy meetings, some of them. I was on their advisory board for a while. Oh, okay. Now you have a question about AI. I don't know. I'm not an expert in AI. Can AI possess emergent properties? People, garbage in, garbage out. So AI needs to be done well. I mean, but the big breakthrough in our AI, as everybody knows, is the solving or largely solving the protein folding problem, which was the breakthrough of the year in Science magazine last year. There's all this talk about AI taking over the world. We got to make sure that doesn't happen because it could make all kinds of mistakes. It's got to be well controlled by humans. But AI may be very useful for understanding emergent properties. How maybe AI applied to that minimal cell. It can't just be the minimal. You got to take that minimal cell and do lots of experiments and then have AI try to make sense of some of that stuff. If you do the right kind of experiments, you have to have the right kind of data going in to get good data out. In the case of the protein folding problem, they had the three-dimensional structures of 100,000 or so proteins. That was wonderful data that AI harvested in order to predict how a new protein would fold. And it's that's one of the big successes of AI in science. And it's also being used effectively for image processing. But I'm not an expert in AI by any means. So, but it is a tool that can be used in science. And I hope it won't take over the world. It would be a very, very awful world if it does. I see we've reached the end of my time.
Yeah, we have one more question in
okay,
the chat,
one more.
if you have time. Yeah. What will be your favorite core topics for the biology course 1?
Well, core topic, yeah.
Yes.
Well, I would only be able to. I would try to get the people to understand the cell, not the details, but the cell as a collection of catalysts that make a new set of catalysts. That's really the essence of a cell, right? It's such a very complicated set of catalysts that take food out and put out waste and convert that food into a new cell. I mean, the cell is really the most amazing thing in the universe. If you ask somebody, could you make such a thing? Most people would say no, because we wouldn't exist to ask that question. And we still don't understand it, but the fact that it exists is so amazing. And yet I watched my grandchildren take biology and learn about cells, and they said the cell is the most boring thing in the universe. It's a box full of named parts. It's so distressing. So I actually try to do something about that. I've been involved with an organization, NGO, that came out of the Academy called the Strategic Education Research Partnership, SERP, S-E-R-P. And if you Google SERP education science, you'll find a little, we made some units for 12-year-olds to teach cell biology. And the unit that I worked on is based on having students analyzing thinking about what would it take to make a self-replicating robot, right? There's a bunch of cartoons, and so they have to. They eventually, you want them to say, well, you need material, need a place to put waste, you need information, and you need energy, and then it goes on from there to use a brewer's, yeast from a grocery store, to make the analogy between the robot and the yeast. So we tried to do that in that little exercise. At any rate, I hope all of you guys will keep on pushing for a young academy in Serbia, and I wish you all the best of luck in the future.
Thank you. Thank you. It was really our pleasure having you today with us. Thank you.
Well, thank you. Take care. Bye.